Hello, welcome in. So good to see you. You probably don't recognize me because I'm not wearing a tropical shirt. You're like, who is this? And what have you done with Professor Zook? There's a story behind it, and it's just that I've been very busy today. I was running errands, and so it's a little rainy. So this is my errand running outfit, and I have more errands to run. So please bear with my less than tropical shirt today. This is a very, we'll call this a West Coast shirt. Didn't want to throw you, confuse you at all. Glad to see you here. This is a very different kind of presentation you're probably used to with me, but I'm just going to, basically try to answer all the questions that I thought were kind of unanswered in a very clear way. And the reason I thought they were unanswered is because I've been very unsatisfied with the information I've heard from news sources, and I also, hear things from like my mother when I talk to her, and she's like, I have no idea what any of this means. And I'm like, okay, if you don't know, then probably a lot of other people feel the same way. So we're going to have, we're going to go all over the place. And so it's a town hall. Here's the way it's going to work. I'm going to talk for about 40 minutes, possibly 45. I got a presentation prepared. As I'm presenting, as I'm talking, if you have any questions about what I'm talking about, or if you just have a question of your own that you wanted to ask, knowing that I might get to it in my presentation anyway, the easiest way to do this is to put the question in the chat part of Zoom. So just write it on in. Anytime I'm talking, it won't distract me in any way. And then after I finish talking, I'll close my slides, go back to full screen, and start talking about addressing the questions. And I know someone will at least ask this question, which is, where are you today? Well, I'm in Iceland, which has done, by the way, a very good job with COVID, unlike us. So there's my introduction. Again, thanks for being here, everybody. Glad to see you. I'm going to go ahead and get my presentation started by magically calling up my slides and getting started. So here we are, going viral, an OLLI Town Hall. So here we go. Now, to be honest, the kind of thing that, remember this image, because we will all, in 10 years, you'll see an image like this, you'll know exactly what it was. You'll know exactly this moment in time. But this is also an image that I don't even have to really tell you what it is. You all know what it is. And it's just a sign of the moment. And I thought of this as soon as I thought of doing this today, because this should never have happened. We should never have had empty shelves of toilet paper. And what drives this is fear, and the fear is driven by what people don't know. And so this was the image I had in my head when I thought, let's do this town hall. And Michelle asked me to do it. I was like, happy to do it. So here we go. Oops, there it is. Some of you, I can't even get out of my, there we go. What happens sometimes, for those of you that have not been through this before, is Zoom's security feature throws me out sometimes and I have to get back in. So not to worry, here we go. Sorry for the small distraction. There's a skill set that I've learned through this pandemic, one of which is how to navigate Zoom in ways that I never thought I would ever have to do. Okay, now we're back up, and voila. So here we go. First of all, what is a virus? A virus is a very simple thing that can do a lot of damage. Almost all viruses consist of two things: a protein capsid, which is like the shell it's contained within, and a nucleic acid core, which can come in 4 different formats. Viruses can also be what are called naked or enveloped, which simply refers to whether they have an additional layer they're contained within. They come in lots of different shapes, and the nucleic acid on the inside takes one of 4 formats: single-strand RNA, double-strand RNA, single-strand DNA, or double-strand DNA. And that actually matters. I'm going to basically focus on the coronavirus today for obvious reasons, but what I want to start with is, it's easy to get in the mindset of thinking, wouldn't it be great if we could just wipe all viruses off the earth? And the reality is that's never going to happen. The reality is we have to learn how to live better with viruses, because among other things, there are actually more viruses on Earth than there are stars in the universe. There's probably just as many viruses as there are, living things and cells on Earth. So we're not ever going to get rid of all of them. And in fact, as you'll find out, we probably don't want to do that either. This is going to be interesting. Okay, one more time. Let's try this again. Was thrown out of my slideshow yet again. Okay. This usually settles down after a few minutes, because what happens is anytime there's a change, Zoom decides I'm a security threat, and I'm not, I swear. There we go. Are viruses alive? Actually, viruses are interesting because they give us this profound question of what it means to be alive. So viruses are not alive, but they're not also dead. The fact is, a virus can just sit there for, quite frankly, 1 million years in, like, say, the Antarctic until climate change releases it. And if it can find a host, that's when it starts replicating. That's the whole purpose of a virus, is simply to replicate itself. It does so with very different methods. There's lots of different viruses. They all have different methods of getting into a cell and manipulating the cell to kind of make more viruses. A key thing to understand, because we're going to talk about it today, is something called either tissue tropism or cell tropism. And all that means, it's a fancy way of referring to viruses have to find a cell that will accept them, that they can sneak into and basically hijack. And it turns out most viruses spend a lot of time trying to find that tissue or find that cell. I'm sure you've all heard people talk about how, this virus jumped the species barrier, and I'll talk about that today. A lot of that has to do with a virus basically altering itself so that it can find a way to get into a different type of cell. Now, again, we have lived with viruses since the start of everything. And you know how we know that? Because if you see that pie chart, 8% of human DNA is actually viral DNA. We wouldn't actually be who we are without our interaction with viruses. We wouldn't have evolved to be what we are. So, without viruses, we wouldn't be the human beings we are now. So again, it's about learning how to live with viruses, not about thinking, I wish we could get rid of all of them. Okay, so let me try this again. This is the first time I had to do this like every 10 seconds, but okay. One more time, getting in and out of these slides. I don't know, like I said, it should settle down. Here we go, one more time, and hit play, and there we go. So what does a virus do? What's the life cycle of a virus? It's pretty straightforward. It attaches to a cell. That's that cell tropism. It gets inside the cell. It takes off its coat, which is its, kind of protein outer coating. It then engages in two things. First of all, it kind of shuts the cell down and manipulates everything the cell does to make it work for the virus. That's called early protein synthesis. And then after that, it makes the cell work to reassemble more viruses, and that's late protein synthesis, which then assembles the virus, and then it releases itself. And most viruses, when they leave the cell, they destroy the cell. It's not the same thing as a cell self-destroying, and that's important to understand how our immune system reacts. But most viruses replicate in what's called a lytic cycle, which means when they break out of the cell is killed. Not all do. There are some latent infections which go with something called a lysogenic cycle, but the coronavirus is certainly a lytic cycle type of virus. So it basically kills the cell when it leaves. Now, that's not the important thing to understand about the coronavirus. It's really just about understanding, let's get to know viruses. So why do viruses exist? Well, viruses exist because life exists. Like I said, we just have to kind of learn to live with them. They were part of the fertile soup of biogenesis. When life started, viruses started at the exact same time. And that's why, as I said, 8% of human DNA is actually viral DNA. Everything finds a way to survive. And so it's easy to think of viruses as these evil kind of zombie-like things that are out to kill us. But they are a byproduct of everything that made life in the first place. By the way, you may know that the Nobel Prize this year went to one of my Berkeley colleagues for the gene editing tool CRISPR. What you may not know is the inspiration for CRISPR, the gene editing tool. All of it was learned from a bacteria. a bacteria that had this amazing ability that every time it was attacked by a virus, it could edit the gene of the virus after it fended off the attack to make sure that if it ever met that virus again, it would know what to do. And it was like, wow, how is this bacteria doing this? Well, that inspired what may be the most powerful gene editing tool we've ever had. Sorry. Every time someone gets let in, this is the issue. So let me just keep cycling through with the greatest of speed. And doop, doop, 3, and one more click, and we're good to go. So now let's focus on the coronavirus. What is a coronavirus? It may not look like it from that picture because that's a 3D picture, but if you see the 2-dimensional picture, it's named a coronavirus because it looks like a crown, which is the word corona in Latin. You probably thought it was named for the beer. And if you heard the story that people weren't drinking Corona beer because they thought it might be attached to the coronavirus, that is actually true. People actually were like, don't drink Corona because it might have the virus in it. It's like, yeah, nothing tastes better than a brewed virus. It looks like that. It's spike proteins around an outer capsid. So there you have your capsid. Inside is the nucleic acid. And it's those spike proteins that we're going to focus on a little bit today, because those are the things everybody's worried about right now. And I'll tell you why. By the Baltimore classification system, this is considered what's called a class 4 positive sense single-strand RNA virus. In the same family as coronaviruses are the picornaviruses, one of which is polio, for instance, the polio virus, flaviviruses, togaviruses, and coronaviruses. And coronaviruses are actually the most complex, all of them, because they have unusually long genomes. As you'll find out, that's actually good news for us. And I'll explain that in just a second. Just another thing to understand, the minimal amount of a virus needed to basically induce the onset of illness is called the infectious dose. And this is one of the many things we don't know about the coronavirus, is we don't yet know what the infectious dose is. And that's one of the reasons you hear what sounds like contradictory information, like how much do you have to inhale, big droplets, small droplets. And the reason is, quite frankly, we just don't really know as much about coronaviruses as we would like. So it sounds contradictory, but the reality is we need to know more about coronaviruses and how they spread to really have a definitive answer. And that's why we don't yet have an answer that says, for some viruses, we know exactly what the infectious dose is, but we don't know for coronavirus, but we're knowing more every day. It's one of the reasons why wearing a mask is always just a good idea, as is finding the most interesting way to get through a slideshow like this, like just like a virus. A virus replicates itself. I'm replicating my slides every 2 seconds. All right, one more time. Like I said, this should settle down shortly. So how does the coronavirus work? The coronavirus works, its cell tropism happens to be, for the most part, respiratory epithelial cells. And what that means is that the cell that it found a way to attach to with its spike proteins and found a way to get inside happens to be respiratory epithelial cells. That's actually, it's good news and it's bad news. It could have been any cell, like rabies, for instance, finds brain cells. Coronavirus, respiratory epithelial cells. After penetration into the cell and its uncoating, the single-strand RNA then morphs into 10 subgenomic RNA strands and starts the replication process. Now, that's unusually complex for a virus. The complexity, the transcription regulating sequence of coronavirus, which again is unusual, all that means is that basically the coronavirus is one of the few viruses that basically has a complex sequence. It may sound like, oh no, it's going to keep coming back and it's going to do terrible things. And yeah, it might come back, as we'll talk about, but this is good news for reasons I'll explain in just a second. Now, the spike proteins, what they do is they get inside, you inhale some. The spike proteins, their matching thing that they can attach to that's on this respiratory epithelial cells, what they attach to is a transmembrane protein, which is referred to as ACE2. And I'm going to talk more about this because some of you may have heard of what are called ACE inhibitors. And it's like, is it related to that? And the answer is yes. ACE2 is a transmembrane protein. Membrane is the cell membrane. So it means as soon as the coronavirus finds that ACE2 protein sticking out, which also it's on the outside and the inside, it attaches with one of the spike proteins and immediately induces endocytosis. And all that means is that's when it starts pumping its nucleic acid into the cell and making the cell basically transform into its host. So ACE2, what does it stand for? You can see it's the angiotensin converting enzyme number 2. Sometimes it's called human for the H. You'll see it says ACE2 or HACE2, and the little h is for human. And that's important to understand because it turns out the reason the coronavirus might have skipped species is because ACE2 cells are found in other species. So human ACE2 was not the usual cell tropism for this virus, but ACE2 cells in, say, a bat versus a human are at least relatively similar. Now, here's another thing you should know about ACE2 cells. Even though they are found most abundantly in lung type 2 alveolar cells. Now, again, if you don't know what that means, all it means is that's the part of the lung where the gas exchange actually happens. That's where the blood says, here's some CO2 that we don't want, and give me some oxygen. That's where it happens. And that is exactly why one of the first things we associate with the coronavirus was the inability to get enough oxygen, because it was basically, remember, it kills the cell after it, lytic means it kills the cell. So what was happening is the alveoli in the lungs where the gas exchange happens were being killed off. And that's why it was so hard to get enough oxygen. Also understand, and this is also important, I'll talk about why ACE2 cells are found most abundantly in the lung and the alveoli, but they're also found in the heart, in the arteries, and the intestines, and other organs as well. So, that's important because you'll understand, why there was the symptoms are always, they seem like a mystery. Like why are some people having this problem, some people are having this problem? And I'm like, it's actually, once you understand how the coronavirus works and what cell it attacks, it makes sense, as you'll see. It makes sense. Sorry, just this is going to be. I'm really getting my Zoom skills up to warp speed today because of this. Anyway, sorry for the inconvenience. It's not bothering me, so hopefully it's not bothering you. This just happens sometimes. Next. Okay, so where did it originate? There are different theories, but here we go. So we know it's what's called a zoonotic virus that jumped the species barrier. And that sounds very frightening, but what it really means, and you should be able to just, kind of follow this along, all it means is that spike of the coronavirus, say from a bat or from some other species, it means that spike mutated. And I'll talk about why viruses mutate in just a second. It mutated and led, in essence, to human cell tropism. So had it not done so, we could have inhaled, a gallon of it, and it wouldn't have been able to attach to human cells. So we know that coronaviruses thrive among bat populations. It seems to thrive among bats, even though it doesn't necessarily sicken bats. That happens frequently, as with swine flu. Pigs are able to catch human flu but not be affected by it, but it means the virus is able to stay inside their system and actually replicate, even though they don't show symptoms. The reason that we don't know a lot about this virus, the reason that we're like, why don't we know exactly how to stop the coronavirus, is because research on coronaviruses didn't really start until very recently. It was when the first SARS epidemic, the sudden acute respiratory syndrome COVID-1 virus, broke out in Asia in 2002, that researchers thought we need to figure out a coronavirus is. It wasn't considered a primary focus of research because it wasn't considered anything that humans had to deal with. And then suddenly in 2002, we had that first SARS epidemic. Now, the first SARS epidemic had about a 10% mortality. Researchers started looking at coronaviruses, and then suddenly we had what was called MERS, which was Middle Eastern Respiratory Syndrome. That had a 50% mortality rate, which was a huge wake-up call. So SARS, COVID-2, SARS, basically coronavirus, CoV stands for coronavirus 2, which is what we're now in 2019. The reason we don't know more than we'd like to is because even though we've had, say, 17 years to do research on a coronavirus, viruses are complex, and the coronavirus is among the most complex. So for instance, you can see this chart here going from bats. There's different theories. Did the coronavirus go from bats straight to humans somehow? Or did the coronavirus go to an intermediate species like a pangolin, which, by the way, is an illegally trafficked animal, gets illegally trafficked into China? So it's a species that was out of its natural environment, so it becomes the vector of the virus. It's one more reason why the illegal wildlife trade is something the world really needs to take seriously. Another possible intermediate species was the civet cat. And then eventually, if the civet cat gets this and the virus is able to replicate and mutate, and you get a civet cat in, say, a market, and somebody buys it as a pet or eats it, it can jump the species barrier. So it's not necessarily easy to do. A lot of things have to be in place, but understand it's always a possibility, and that's all it takes is a virus to change its cell tropism just a little bit, and for us to come into contact with animals that we don't usually come into contact to all the time, and we can get a pandemic, such as the one that we have. Okay, gonna keep doing this apparently slide by slide. Okay. Anyway, almost up to speed here. Here we go. One more click, and here we go. I can move forward again. So why are there so many variants? And this is the thing that's scaring people right now, and understand that variants are a natural part of the process. Variants are how a virus can jump the species barrier. And the fact that we're seeing variants is not surprising, but it also is a reason we should still be careful. The variants you're hearing about come from something called misincorporation. Misincorporation is when a virus replicates, basically it's DNA, it's RNA, it's single-strand RNA gets replicated into more single-stranded RNA using the same translation transcription process of the human cell. Imagine if I said, type a sequence of 100 numbers a billion times in a row. You'd probably make some typos. After a while, you're like, okay, and you make some typos. Well, that's what happens here. You've got billions of replications, and sometimes things go wrong. These misincorporations, when, say, different parts of nucleic acids come together in a way that weren't supposed to happen, just a mistake in the transcription process, that's what creates random mutations. It's just a part of the evolution of anything. Human evolution has variants. Now, coronaviruses, you heard me say that they're more complex, and that's a good thing, because it turns out coronaviruses actually have something most viruses don't, which is the ability to proofread the replication process. It's a complicated procedure, but all you need to know is that the coronavirus can actually detect and remove most misincorporations, which means we get far fewer variants than we would get with another virus. And that's why I said it's good news. Even though we have these new variants, the coronavirus is one of the few viruses that has this ability to kind of check whether the replication process went as planned. And that means we're going to get fewer variants. Now, but remember, like with human cells, basically 1 in a billion replications of DNA creates a misincorporation. Now, granted, about 6 billion happen at any given time in the human body, so 1 in 6 has a chance of at least making one mistake. So this is a natural part of all cell growth. It's just a natural part of the evolution of anything, including a virus. So what does this mean? So misincorporation is going to happen. It's just going to happen more slowly because of this peculiar feature of the coronavirus. When you see all these numbers about B117 variant and this and that, there's a reason for it. The thing to pay attention to is when you see VOC, that means variant of concern, which means doctors are saying there might be something to this we need to worry about. But let me tell you about what that worry really is. So there are different ways of referring to these variants. B117 is basically a reference to its phylogenetic heritage, like what came before it. 201/501Y.variation1 refers to its genetic location. The one I like best is actually the last one, which is N501Y, and all it means is that at position 501, so they map the virus at position 501, because of a misincorporation, the regular amino acid that was on the spike protein, which is asparagine, was basically replaced with tyrosine. Just for bonus information, if you've ever seen like, the flu information, H1N1, H5N5, you're like, what's going on here? Again, if you know what these labels mean, it's pretty straightforward. And just knowing that helps you kind of be in control of the information. So when the flu shots, when they design these flu shots, what they have to predict is something very complicated. Influenza viruses also have spike proteins they use to attach to cells. And the spike proteins are basically composed of either hemagglutinin, which is H, or neuraminidase, which is N. And you can see there are 17 types of hemagglutinin and there are 9 types of neuraminidase. And if you're into statistics and things like that, and you figure out how many possible variations you can get with 17 of one thing and 9 of the other, it's a staggering amount. And so what they have to try to predict every flu season is which variation is probably going to emerge. And they can get it wrong. But when you see things like, you're getting the H3N5 vaccine, it's because they've basically used very powerful statistical models to say this is the most likely version that has evolved. And again, why do we have different versions? Because of misincorporation. Happens in all viruses, actually happens in all processes of replicating DNA. So the ones that are most worrisome in terms of these variants would be ones that might alter the shape of the spike protein that's used for cell attachment. So when you hear there's this new variant that makes it 100 times easier to infect you, it sounds very frightening, but now you understand what it means. It just means that the spike protein had a misincorporation, which led to a variant, and that variant is just a lot better at locking onto the cell, the ACE2 receptor. And that's why, so it's not a more virulent virus, it just means it can catch a hold of the cell and replicate itself easier. So it's not a more powerful virus, it's the same nucleic information, it just has a better Velcro, is one way to look at it. That's what it means. Now you're thinking, uh-oh, what does that mean for the viruses? Or what does it mean, what does it mean for the vaccines? I will address that. Let's see if I can get through a slide. Nope, can't advance. I did it once, but it didn't quite work this time. Okay, one more time. And here we go. Not sure why this is happening, but share my screen. There's the slides. Share and go. So why Wuhan? Technically, it could have been anywhere. It just happens that you can jump the species barrier wherever humans come into new environments or wherever other species come into new environments. We know that bat populations naturally harbor the coronavirus, and there's lots and lots of bat viruses in areas just to the south of Wuhan. We also know Wuhan has they're all over China actually, what are called wet markets. Wet markets are a terrible thing. They are live animals and fish in very close proximity, usually in very small cages, which makes them stressed, which makes them more vulnerable to, as you know, with humans under stress, when your immune system weakens under stress. And so you have animals who are more likely to catch a virus, such as a civet cat at a market, and then a human buys it, takes it home as an exotic pet, eats it, what have you, and that's how it can jump the species barrier. So between wet markets, high human population density, and the human encroachment into new environments, Wuhan just was a classic vector for this to happen, but it could have been really anywhere. Wuhan just had the perfect conditions if that's in fact where it started. Unfortunately, the government of China destroyed the wet market and basically sanitized it before any evidence could be collected. So we'll never know for sure if that's where it came from. But the other question everybody has is, what about that lab? What was going on in that lab? So the Wuhan Institute of Virology was doing research on coronaviruses. This we know, and that's not necessarily suspicious because Asia was where the SARS epidemic broke out. And so you have a number of laboratories that are trying to find out more about coronaviruses. So why are we researching coronaviruses? Why are we researching viruses at all? What's going on in these labs? Well, one of the things we're looking at in terms of medical research is, remember that viruses have a specific cell tropism. So if you use the CRISPR gene editing tool to say, edit a gene and cure something like Alzheimer's, you still have to find a way to get it to find a brain cell. You can't just put it in someone's bloodstream and say, I hope you find your way. So what do? Well, In this case, what virus goes to the brain cell best? Rabies virus. So you basically take a rabies virus and you figure out how much can you take out of it so that a person doesn't get rabies, but it still finds the brain cell. And then you put the edited gene back in that replicates a healthy brain cell. And then you inject a person with basically an altered rabies cell. That's how it would work. So that's one thing that goes on in these kinds of viral labs. The coronavirus, as I said, is a relatively new and still somewhat unknown virus. The other thing that was going on in this lab, and this is a very controversial area of research, is what's called gain of function research. And there are some states that actually banned this. Gain of function is where a laboratory intentionally tries to induce, through misincorporation, different variants of a virus to see what would happen. So they basically induce misincorporation, which means they create variations to see if they can find a version that's, what if this protein changed? What if this spike changed? Would it make it more virulent, less virulent? And the problem with that is if you create a version that becomes, say, 10,000 times more virulent, that can be among other things. If it escapes the lab, now you have a pandemic of terrible proportions. There's also suspicion that especially the Wuhan Institute of Virology has what are called People's Liberation Army researchers who are considering gain of function research as a form of bioweapons. So it's very mysterious exactly what was going on. Scientists have not been able to rule it out as a possibility for the source of the pandemic. China is denying this, but as you know, the World Health Organization went to China to do its investigation, and they were not given access to the Wuhan Institute of Virology. And so until there's a transparent investigation, it still remains a plausible explanation of where this all came from. We now know that in 2018, American diplomats warned the United States that they thought that the personnel at the Wuhan Institute of Virology were not trained properly in the security measures of what's called a high containment laboratory. So we do have some evidence that it's plausible, at least, that it did escape from the lab. Other questions. Why do some get so sick when others are asymptomatic? Well, again, let's see if I can do this in a few minutes. Humans have two response levels to any virus. We have the innate immune response, and we have the acquired or adapted immune response. The innate immune response is basically when the body detects anything that doesn't belong there, it basically goes to work and says, I haven't seen this before, it probably doesn't belong here. So we send out, the first line of defense. Macrophages are specialized cells. They eat the germ cells, and then they leave behind these things called antigens, which are kind of like clues, identifying markers of the foreign thing. The antigen presenting cells kind of circulate through the body saying, I found this, and if you find it, it doesn't belong here. So if you're a T cell, you need to destroy it if you see this and if you detect this. Part of that response are what are called helper T cells that help spread the word to nearby cells. Hey, something bad is here. You might want to kind of shut down your cell boundary. So these cytotoxic T cells, that means that these cell-killing T cells that are looking for infected cells, for instance, part of this response involves something in the immune system called a cytokine. And this, you've probably heard the phrase cytokine storm, and cytokine storm is different than what's called cytokine release syndrome, which is a long-term syndrome, which can still be triggered by the coronavirus. But the main thing to understand is that what happens here is that sometimes the body goes into what you might call overdrive. It basically sends out cytokines, such as there's a cytokine called interferon, which basically tries to block the ability of a virus to attach to a cell. There's another one called, or it's not a cytokine, but it's another particle or another enzyme, not an enzyme, a protein called neutrophils. And all of these are involved in the inflammatory response. And the inflammatory response, if you get a mosquito bite, you get a bump. It's your defense system trying to prevent whatever you got from the mosquito bite from getting anywhere else. And inflammation is part of the response. Well, if your body goes into overdrive, that inflammation can spread throughout your body, and it results in things like tissue damage in the short term or the long term. So when people, you've heard of people getting organ failure and so many things, it's because the immune system kind of went into overdrive. And it doesn't always happen just in relation to viruses, but this is one such response that some people have but not everyone has. And you're thinking, well, how do we know who's going to get what response? Some of it we don't know, but at least part of it we do, and I'll explain that as well. So if you don't know this, by the way, just a little factoid, COVID stands for coronavirus disease, in case you're wondering why we had COVID from 2019, COVID-19. So this might help explain this. And again, some of you may have heard of ACE inhibitors. Here's an example of one, ramipril. So remember that the coronavirus latches on to ACE2. That's its cell tropism. What does ACE2 do? So again, this is the angiotensin converting enzyme number 2. What does it do? It moderates and mitigates the effects of angiotensin 2. Okay, what does angiotensin 2 do? Well, it increases blood pressure, and by doing so, it exacerbates inflammation. You already know that inflammation is part of the problem when some people get so sick. So between destroying lung cells and not getting enough oxygen, we also have potential organ failure due to just inflammation of everything throughout the body. So ACE2 is good because basically it will reverse the rise in blood pressure from angiotensin 2. However, when SARS-CoV-2 binds to ACE2, remember, it destroys the cell, so it knocks ACE2 out of commission, which is one of the reasons why early on we knew that one of the populations that were at great risk for the sudden onset of rapid illness were people who had hypertension, because this was going to make that situation that much worse, because ACE2 can help regulate hypertension. We also know that ACE2 cells are not distributed exactly equal in any two human beings. So if you have more ACE2 cells in this particular organ or in your brain or elsewhere than someone else does, or in certain parts of your respiratory tract, you may be more susceptible to infection and to, well, being what's called a long hauler, for instance. So between the gas exchange inhibition, hypertension, other things that can happen, heart attack, stroke, even organ failure. And this is all because of knocking out this ACE2 when it attaches. And so now, when you've heard these stories of people who would get coronavirus, even young people who were dying of strokes at the age of 28, it's like, how is this happening? Well, it's because we have ACE2 cells in the brain, and if the virus finds its way to an ACE2 cell, and basically you have a massive change in blood pressure, you end up creating the classic circumstance of inducing a stroke. So it seems like it's just this virus that attacks all these different ways, but if you understand how it works, you can understand, well, all the things you've read about. It's not quite as frightening anymore. So that takes us to the vaccines. How do the vaccines work, and what's going on with the vaccines? Well, a vaccine works by introducing pieces of SARS-CoV-2 or SARS coronavirus 2 to induce the adaptive immune response. Why do we need the adaptive immune response? Because our innate immune response is not enough. For some people it is. Some people get COVID and they get over it, but not everybody can. And the problem is even people that can get it and get over it are keeping the virus in circulation. So that's why we need this secondary line of defense to get our adaptive immune system to back up the innate immune system. It's classic how most viruses work. You introduce something into the bloodstream that produces antibodies that attack the antigens. You get the T cells worked up. The main thing is you get the T cells attacking whatever you've introduced in the bloodstream, and then we get these memory T cells that basically say, aha, I will remember anytime I see these antigens. So if you get it in the future, I know to attack it. And the process takes a couple of weeks, and that should sound familiar, because after your second shot, they say wait 2 weeks. There are different types of vaccines that make this happen. There are mRNA vaccines. And by the way, I will share these slides with everyone also, so you can have a chance to refer back to them. mRNA vaccines basically introduce an RNA sequence that builds a harmless protein that, however, is also part of SARS coronavirus 2. Viral vector vaccines basically do the same thing, but they use a different virus that's related to coronavirus to deliver the payload. And then protein subunit vaccines basically just introduce the particles directly into the bloodstream and hope that your immune system finds them. So what are the vaccines that we have right now? Well, here's what we have. We have two that are mRNA vaccines. The Pfizer one and the Moderna one are both mRNA. So they introduce something, basically your cells produce this protein that your body will recognize as being something that doesn't belong there. That protein is also in every variant of the coronavirus, and therefore it will take care of it. The efficacy is very high, 95% for Pfizer, 94.1% for Moderna. The Johnson & Johnson vaccine is a viral vector vaccine, and you'll see that its efficacy is much lower, 66.3%. So why would we even want that vaccine? Well, because the Pfizer and Moderna vaccines are very expensive to store, and they do need 2 shots. So it's a trade-off between Johnson & Johnson's, which can be stored, basically refrigerated without the high energy, the medical grade freezers you need for Pfizer and Moderna. So logistically, Johnson & Johnson's is much easier. So it's a trade-off between efficacy versus ease of transmission and ease of getting the vaccine. So far the variants are, variants of concern, but the vaccines still seem very effective against them. And the reason is because the proteins involved in the vaccines that spark the immune response are not spike proteins. That means if the spike protein has variations, so far it won't matter, because the mRNA shot that goes in basically is creating a protein that's unique to the coronavirus, but it's not a spike protein. If it were a spike protein, that would make us worry a lot more. So it means that the spike proteins can have all the variations they want, but we still seem to be immune because the protein that we're developing the immunity with is something that is not basically going to be variated or misincorporated out of existence. So we have a whole new virus to deal with. After the vaccination, can we return to normal? Texas thinks so, but they're wrong. Not yet. And here's why. Even though I'm getting my 2nd shot next Friday, and a lot of people are getting vaccinated, and that's great. But the thing is, the more the virus circulates, the more variants it will produce. And that raises the chance of the possibility that one of those variants created by misincorporation could be the thing that escapes the virus. And if it can escape the virus, it'll replicate out of control, and we'll be right back into a new pandemic. So we still need to be very careful until we know we are close to, or we've achieved, what's called herd immunity. And that's going to take several months, quite a few months actually. Vaccination does reduce transmissibility. That's the good news, because it takes care of how much is in your body. So even if you catch, if you've been vaccinated, if you inhale another coronavirus infectious dose, it seems that it minimizes your ability to transmit it to other persons a great deal. And that's good. But remember, it reduces, it doesn't eliminate it. And that means we should still wear a mask. We should still be careful. Higher vaccination rates should accelerate the return to normality. So this is the plan. Wear a mask, still be careful. Higher vaccination rates, the higher those get, the better off we are. Another question people have, did we overreact? I hear this all the time. This is basically just the flu. Look at the mortality rates. No, not just the flu. Remember that we don't know a lot about the coronavirus. And MERS, for instance, had a mortality rate of 50%. We didn't know when this started. If this had a 50% mortality, this would have been, a magnitude 100 times more frightening. We didn't know what this coronavirus is going to do. So it made sense to kind of shut everything down until we knew. So that initial uncertainty meant that we had to react quickly, and that was the right call. Also, we knew that people could get this, just like animals can get the virus and not have any symptoms, but they can transmit it. So we knew that asymptomatic transmission was also possible, and that was going to hit vulnerable populations very hard. Also, the focus on the mortality rate is the wrong thing. Like, well, 500,000 people are dead. That's like any flu season. Well, number one, we didn't know about the coronavirus, where it was going to go. We didn't know how quickly variants would evolve. But remember, even if we had no fatalities, when we have 0% capacity in an ICU, we are unprepared for anything. People who have other things happen to them and they need an ICU, if our healthcare system is overwhelmed, so focusing on mortality is the wrong thing. And by comparison, you'll see, it's like saying September 11th killed 2,977 people. Why are we so afraid of terrorism? Our chances are so small that it's going to kill us. So let's get rid of security. Nobody would want that. People would say that's ridiculous. So no, we didn't overreact. And you know, continuing to be cautious is a good thing. So we brings us to, you know, kind of our end here. What does the future hold? Well, how's this going to play out? Well, first of all, I have to say we could have done this so much better. And I'm not going to blame one party or the other, because I think in many ways, part of the tragedy was this was leading up to an election. So both parties were using the coronavirus for electioneering, and that really made me angry. We definitely need a better plan, a plan that is basically completely independent of whichever party controls the government at any given time. I hope we can all see that at this point. We got lucky, quite frankly. This was a dress rehearsal for the next pandemic. Keep your masks, don't throw them away. And if we can figure out our mistakes this time, we'll do a better job next time. There are so many things we have learned from this in terms of, say, what's called intersectionality. We understand so well how, you know, we'll keep understanding better how the pandemic hit vulnerable populations, different racial groups, how it affected different people because of their position in the economy, so many things. But here's the thing, and if you don't know what this picture is from on the slide, this is from something that happened right here in San Francisco, where people jumped into an Uber and for some reason thought it'd be very funny to cough all over and pepper spray the driver because they thought he was Asian. He's from Nepal, but it's part of those attacks. And so, you know, my biggest fear is that we have this ongoing epidemic, two of them actually. One is human idiocy, and the other is amnesia. I'm so afraid we will not remember what we all went through, because the next time it hits, hopefully the response won't be quick, buy 47 tons of toilet paper. Hopefully the response will be, we can do this. We know what we have to do, and we know why we have to do it. It's not because a face mask is tyranny and I'm being oppressed. It's because understanding how viruses work and that we have to find a way to get through them is what's going to make us either avoid or at least get through the next pandemic. As I said, I put all this together because I know it was a lot. It's being recorded. You can go back and watch it as well. And also I will share these slides. I'll send them over after I finish today. But my impression is I've talked to so many people and everybody feels a little bit lost, and it feels very scary not knowing, you know, what does a variant mean? Well, I thought, let's just talk about what a variant means. And is it something to be concerned about? Yes. But is it something to think, oh my God, a variant means the vaccine won't be effective? The worst case scenario with the variants, if they do affect the spike proteins, it means we're all going to need a booster shot, a third booster shot. That's what it means. So that's my presentation for now. And I see there are some questions in the chat. So let me go and see what's going on with that. Assuming one of the things I didn't put in my presentation that I will probably talk about, and there might be a question about it, is I can't tell you how unsettled I am by violence against people of Asian descent. It's the toilet paper response. It's this irrational fear of wanting to either hoard things or harm things that have zero relationship to the coronavirus. It's just created by, it's a violence created out of complete ignorance. So here we go. Let me look at some of the questions here. Let's see. Okay, people on time. Most of the comments are about that. Yes, it's being recorded. There we go. Goes to Caleb. Here we go. By the way, somebody's asking, what type is the AstraZeneca vaccine? I don't know. I didn't have a chance to look it up. I believe, however, it's an mRNA one, but I'm not sure on that. If I can, I'll do some research afterward. What I can do is update that slide, you know, and I can add a fourth thing and talk about the AstraZeneca slide as well. So someone said, I thought you said people who had the virus and got over it might still transmit it. Why is that? So people who got over the virus, it depends. It's all an issue of timing. Like my niece, for instance, got COVID-19 and she recovered. And so she basically is now donating her plasma, which helps in the manufacture of some of the vaccines, for instance. But again, we don't know as much about coronaviruses as we want. And we know that, for instance, it is possible to catch it again. Now, this is before we had the vaccines. Usually once you get a virus, your body, your innate response and your adaptive response, can make sure you don't get it again. But with influenza, same thing happens. The problem is influenza is a virus, for reasons I explained, that gives us a staggering amount of variations. So we don't yet have a universal flu vaccine, and that's why you can get the flu over and over again. And so coronavirus isn't quite as bad as that, but it's possible, if you're asymptomatic especially, that the virus is basically replicating. You're not showing symptoms, but you can transmit it to others. And even if you've been vaccinated, even though that ability plummets down to about 5% of what it was, remember, that's not 0%. And this is why, it's not uncomfortable to wear a mask, for even though I'm going to get my second shot mid-April, I can say, yay, it should all be there. I'm still going to wear a mask. It's just not that hard to do. I'll give it a few more months. I'm concerned and alarmed. I see these pictures of spring breakers in Florida lying together, and it's like they're thinking, well, I'm young, my immune system can handle it. It's like, that's not the point. The point is that's keeping the virus in circulation. The more it circulates, the more likely it is through the process of misincorporation and evolution that we're going to get a variant that these vaccines may not be able to handle. And that's going to hit vulnerable populations, and even in people that don't consider themselves vulnerable. As you know, there were young people who died of strokes. There were young people who died of heart attacks and organ failure because their ACE2 distribution was such that, and their innate response, their innate immune response gave them the cytokine storm. And how do we avoid that? The best way to avoid it is to keep being cautious, being responsible. This is the number one thing right now. So, and the thing is, I know I'm talking to the wrong crowd, because you're OLLI folks and you're the cool crowd. You get it. But if somebody else asks you, or somebody's like, I am not wearing my mask today, you can say, you need to understand misincorporation. And they'll say, what are you talking about? And you can tell them all about misincorporation and why they should still wear a mask. So why does the Johnson & Johnson vaccine have a much lower efficacy? It's because of the delivery mechanism. The trade-off is the viral vector format doesn't create the same response as the mRNA. The mRNA vaccines are far more effective, but they require 2 injections, and I got my first injection at the Coliseum, and you can see the medical grade freezers. I mean, they make this massive loud noise, and they're enormously expensive to buy and to run. And so the Johnson & Johnson vaccine, because it requires just simple refrigeration, it can be transported much more easily. There's less of a problem if places that don't have medical grade freezers can use it, but the viral vector, the method of introducing it into our bodies in such a way that the virus basically replicates. It's a harmless, it replicates basically the proteins. mRNA is directly replicating the protein that our immune system's response to. The viral vector uses this virus, and it just turns out our bodies react less vigorously to that than the direct mRNA. So it's a trade-off between efficacy and the cost of storage and transportation. However, the idea, 66% sounds like there's a lot of people saying, oh, I got the cheap vaccine. 66% is still very good. And it's highly likely Johnson & Johnson will have a booster at some point anyway. So if you end up with a Johnson & Johnson, don't think, oh no, I'm only 2/3 protected, unlike the 95%. The key thing is that number 66% is based upon the ability to catch and also transmit. And it seems the Johnson & Johnson vaccine seems to be more effective than 66% in terms of preventing catastrophic infection. So it's not like it's not like you're playing the Vegas odds. Because remember, the vaccines don't completely eliminate the possibility. Just like the flu vaccine doesn't say you'll never get the flu. It just means if you get it, you're highly unlikely to get it catastrophically. So even the Johnson & Johnson vaccine, even if you get an infectious dose of coronavirus, SARS coronavirus 2, the Johnson & Johnson vaccine shows a much higher efficacy of preventing catastrophic onset. So you might have a higher chance of getting an infectious dose and your body has to react to it. But it's not a trade-off of I'm only 2/3 protected. The main thing you want is protection against catastrophic onset, and all three viruses are very good at that. Okay, let's see. Part of the problem with dealing with cold is, of COVID is the appalling state of scientific illiteracy in the US. How can we improve this? I wish I had an answer for that. I mean, this presentation, this is less than an hour, and this to me, I tried to think of all the essential questions that have a relatively straightforward answer. And even governments are terrible at this kind of stuff. It's just like, you hear, you don't need a mask. Oh, maybe you do need a mask, or a mask is bad. And it's like, figure this out. So scientific literacy is quite frankly, literacy in general, cultural literacy, just knowing things, having the decency, when I see the picture of those women coughing all over a cab driver because they think it's funny, I'm just like, there are so many levels of ignorance. There's just so many levels of things. And I'm like, where do these people come from? So you're right. If we can improve scientific literacy, if we can improve the educational system, that's going to help us too. I mean, the payoffs of improving education are nothing but positive. And part of that is convincing taxpayers in places like Texas, where I have family, that higher taxes is not socialism. Higher taxes to fund education is called good for all of us. It's an uphill battle, but it's not impossible. And quite frankly, one of the reasons I'm here today is because, Michelle was kind enough to say, let's do this. And I was like, I'm happy to do it. And here I am, and here you are. And this kind of thing can happen all over the place. So I wish I had a straightforward answer, but let's hope we do a better job next time. Why didn't the CDC mandate masks right away? Again, I knew masks would be essential because I do a lot of research in Asia, and in Asia, anytime anything, Asia was where the SARS epidemic started. And everybody knows a mask is just a default option. It doesn't do any harm. The problem is we don't know a lot about coronavirus, and the CDC was quite frankly worried about alarming people because masking is not something that Americans are used to. In Asia, if you get up and you have a sore throat, you throw a mask on. So I think the CDC was trying to not elicit panic. But remember that first picture. What did everybody do? For some reason, everybody thought the one thing you need to survive the end of the world is toilet paper. It's like, really? That's what you went for? How about nutritious food? It's like there was plenty of that. In fact, I had this very funny moment when I walked into a store and all the toilet paper was gone, but all the wine was there. It's like, you have your priorities way out of whack. It's like, if it's the end of the world, I want a good, Cabernet Sauvignon. That's going to be the thing I want to enjoy. So I was like, you people got your priorities way out of line. Let's see. So do you have information about the timing of the second shot? Can it be delayed from the 21 or 28 days to be fully effective? The way it works is actually the first shot gives you a pretty good amount of immunity. So even if you get just the first shot, after about 2 weeks, you have roughly probably 80% immunity. The reason you don't get it all at once is because there's a good chance of creating an onset of getting sick from the vaccine. And I've had that happen before when I was vaccinated once before I went on a research trip. So the second shot is basically really a booster. Your body gets up to one level of immunity, and then the second shot kind of pushes you all the way up to 95%. Technically, you probably could delay it, but the timing is such that the reason the timing is 21 or 28 days is that that's the best way to boost your immune all the way to the full effect of the vaccine. So if you can, definitely stay on track. Like I said, mine's scheduled for next Friday, and I'll be there. I'm going to the Coliseum again. And there you have it. It's 10:59. I got enough time for, let's see. There's actually quite a few questions here. They all kept oh, I see. As I've been talking, lots of stuff's been rolling in. Okay. Better to prevent than alarm. Let's see. One more time. Let's see if I can find one last question here. Well, why did India not have the high rate of infection expected? Well, having spent a lot of time in India, I have a feeling the numbers are grotesquely underreported in India. And there's reasons for it, dealing with remote populations, with the inability to get accurate information in like urban slums. So I have a feeling we're going to find out a lot more in the coming months about this. But again, at the end of the day, what was this presentation all about? Should we be scared out of our minds? No. Should we learn how to live with viruses better? Yes. Should we all get vaccinated? Yes. Should we be careful about how we circulate for the next coming months? Probably. I think it's going to be the end of summer before we can really kind of think, I'm going to go for a walk and not wear a mask. And I can do that. That's fine. And end of summer is fine with me. So there you have it. Now, unfortunately, I did not get to a lot of these other questions, but I do want to say thanks to all of you because we're at 11 o'clock. And remember, this is being recorded. So if you want to go back and review parts of it, you can do that. And I will send my slides over to the OLLI folks and they'll find a way to distribute them so you can kind of thumb through them. I'll try to update the ones about the virus because I had quite a few questions about the other virus that's now available. And I'll do that. But in the meantime, here we are on Zoom. It's so great to see all of you. Everybody's looking great. I see smiles, and when I see you smile, it makes me happy too. Thanks for hanging out with me. We're going to get through this, and I hope to see you soon, maybe in a class as well. So enjoy the rest of your day.
Thank you.
And I will see you sometime soon.
Thank you.
Thank you.